Hello students, this is the fourth video of transgenic plants. Normally transgenic plants we can't explain in one two video. So there are lots of things in transgenic plants. We have to study lots of things. So this is the last video of transgenic plant I am making. So let's begin. First, we'll start with manipulation of ethylene biosynthesis. It has been clearly established that ethylene plays a key role in the ripening of fruits. The biosynthetic pathway of ethylene is synthesized from S-adenosyl methionine via the formation of an intermediate, namely 1-aminocyclopropane 1-carboxylic acid, catalyzed by the enzyme ACC synthase. The next step is the conversion of ACC to ethylene by ACC oxidase. Three different stages have been developed to block ethylene biosynthesis and thereby reduce fruit ripening. First, we will study from antisense gene of ACC oxidase. Transgenic plants with antisense gene of ACC oxidase have been developed. In these plants, production of ethylene was reduced by about 97% with a significant delay in fruit ripening. Second point is antisense gene of ACC synthase. Ethylene biosynthesis was inhibited to an extent of 99.5%. Inserting antisense gene of ACC synthase and the tomato ripening was markedly delayed. Insertion of ACC deaminase gene. Let's know about insertion of ACC deaminase gene. ACC deaminase is a bacterial enzyme. It acts on ACC, removes amino group, and consequently the substrate availability for ethylene biosynthesis is reduced. The bacterial gene encoding ACC deaminase has been transferred and expressed in tomato plants. These transgenic plants inhibit about 90% of the ethylene biosynthesis. The fruit ripening was delayed by about 6 weeks. The strategies 1 and 2 may be referred to as antisense ethylene technology. Let's know about longer shelf life of fruits and vegetables. The spoilage of fruits, vegetables and senescence of picked flowers, collectively referred to as post-harvest spoilage, is major concern in agriculture. This hampers the distribution system, particularly when the transport is done to far off places. The successful manipulations to delay ripening, senescence, and spoilage of various foods will significantly contribute to the appropriate food distribution and thus good economic practices in agriculture. Suppressing the biosynthesis of ethylene appears to be a promising area to reduce the spoilage of fruits, vegetables, and senescence of flowers. The three different strategies to block ethylene synthesis in tomato have been now we discussed. The same approaches in fact can be successfully used for other fruits, vegetables to achieve long shelf life. Now let's know about the genetic engineering for preventing discoloration. Discoloration of fruits and vegetables is a major post-harvest problem encountered in food industry. Chain food additives are added to prevent discoloration. However, these additives may cause health complications in humans. Biochemically, Biochemically, discoloration of fruits and vegetables is mainly due to the oxidation of phenols to quinones catalyzed by a group of enzymes, namely polyphenol oxidase. These enzymes are localized in the membranes of mitochondria and chloroplast. Genetic manipulations using antisense approach to inhibit the synthesis of polyphenol oxidase has been carried. Some success has been reported in preventing the discoloration of potatoes by these strategies. Now let's know about the genetic engineering for flower pigmentation. There are continuous attempts in flower industry to make the ornamental flowers more attractive by improving or creating new colors. Besides prolonging post-harvest lifetime, and the cord flower industry is mostly about 70% dominated by four plants: roses, tulip, chrysanthemums, and carnations. The most common type of flower pigments are anthocyanins, a group of flavonoids. They are synthesized by a series of reactions starting from the amino acid phenylalanine. The color of flower is dependent on the chemical nature of the anthocyanin produced. Pelargonidine 3 glucoside, that means brick red or orange. Second is cyanidin 3 glucoside, that is in red in color, and third is delphinidin 3 glucose, that is blue to purple. Manipulation of anthocyanin pathway enzymes. The enzyme responsible for different reactions in the anthocyanin pathway have been now identified. By genetic manipulations, mutations, it is possible to develop flowers with the desired colors. Most of the flowers, roses, carnation, chrysanthemum, lack blue color due to the absence of key enzyme flavonoids 3' 5' hydroxylase that produces delphinidin 3 glucoside. One company by the name Florizen has genetically manipulated and introduced the gene encoding the enzyme F3' 5' H from Petunia hybrida into the following plants. The world's first genetically modified flower was introduced in 1996. It was a mauve colored carnation with a trade name Moondust. Subsequently, many other flowers have been produced and marketed. Can genetically modified flowers be eaten? In majority of countries, flowers are used for ornamental purposes and not usually eaten. However, in some countries like Japan, flower petals are used for decoration of foods and frequently eaten also. This raises an important question about the safety of genetically modified flowers since they are not thoroughly screened for human consumption. But the present belief is that the anthocyanins are natural plant materials and their consumption may be in fact beneficial to health. Now let's know about genetic engineering for male sterility. The plant may inherit male sterility either from the nucleus or cytoplasm. Cytoplasmic male sterility is due to the defect in the mitochondrial genome. It is possible to introduce male sterility through genetic manipulations while the female plants maintain fertility. In tobacco plants, male sterility was introduced by using a mitochondrial mutated gene encoding the enzyme ribonuclease. The gene encoding ribonuclease, namely Barnes gene from Bacillus amyloidocucens, was transferred to tobacco plants. The ribonuclease is toxic to tapetal tapetal cells and thus prevents the development of pollen, ultimately leading to male sterility. By this approach, transgenic plants of tobacco, cauliflower, cotton, tomato, corn, lettuce, etc. with male sterility have been developed. It is possible to restore male sterility in these plants by crossing them with a second set of transgenic plants containing ribonuclease inhibitor gene. Now let's know about transgenic plants with improved nutrition. Genetic manipulation for improving the nutritional quality of plant products are of great importance in plant biotechnology. Some success has been achieved in this direction through conventional crossbreeding of plants. However, this approach is very slow and difficult, and many a time will not give the traits with the desired improvement in the nutritional quality. So, let's know about amino acids of seed storage proteins. Of the 20 amino acids present in the humans, 10 are essential while the other 10 can be synthesized by the body. The 10 essential amino acids have to be supplied through the diet. Cereals, that is rice, wheat, maize, corn, are the predominant suppliers of EAA. However, cereals do not contain adequate quantity of the essential amino acid lysine. On the other hand, pulses like Bengal gram, red gram, soybean are rich in lysine and limited in sulfur containing amino acids. The essential one being methionine. Transgenic routes have been developed to improve the essential amino acid contents in the seed storage protein of various crop plants. Now let's know about the overproduction of lysine by deregulation. The four essential amino acids, namely lysine, methionine, threonine, and isoleucine, are produced from a non-essential amino acid aspartic acid. The formation of lysine is regulated by feedback inhibition of the enzymes aspartokinase and insert AK and the dihydro dipicolinate synthase. Theoretically, it is possible to overproduce lysine by ab- abolishing the feedback regulation. This is what has been accomplished. The lysine feedback insensitive genes encoding the enzymes AK and DHPDS have been respectively isolated from E. coli and corny, cor, corny bacterium. With appropriate genetic manipulation, these genes were introduced into soybean and canola plants. The transgenic plants were developed produced high quantity of lysine. Now, let's know about the transfer of genes encoding methionine rich proteins. Several genes encoding methionine rich proteins have been identified. In maize, 21 kilodalton gene with 28% methionine. In rice, 10 kilodalton prolamine with 20% methionine. In sunflower seed albumin with 16% methionine. These genes have been introduced into some crops such as soybean, maize, and canola. The transgenic plants produced proteins with high contents of sulfur containing amino acids. Production of lysine-rich glycine in rice. Glycine is a lysine-rich protein of soybean. The gene encoding glycine has been introduced into rice and successfully expressed. The transgenic rice plants produced glycine with high contents of lysine. Another added advantage of glycine is that its consumption in humans is associated with a reduction in serum cholesterol. Hypocholesterolemic effect. Construction of artificial genes to produce proteins rich in EAS. Attempts are being made to construct artificial genes that code for proteins containing the essential amino acids in the desired proportion. Some successful has been some success has been reported in production of one synthetic protein containing 13% methionine residues. Now let's know about the genetic engineering for improving palatability of foods. More than the nutritive value, taste of the food is important for attracting humans. It is customary to make food palatable by adding salt, sugar, flavors, and many more ingredients. It would be nice if a food has an intrinsically appetizing character. A protein monelin isolated from an African plant is about 1 lakh sweeter than sucrose on molar basis. Monelin gene has been introduced into tomato and lettuce plants. Some success has been reported in the production of monelin in these plants, improving the palatability. Now let's know about golden rice. the provitamin A rich rice. About one third of the world's population is dependent on rice as staple food. The mild rice is that the mild rice that is usually consumed is almost deficient in beta carotene, the provitamin A. As such, vitamin A deficiency causes night blindness, is major nutritional disorder worldwide, particularly in people subsisting on rice. To overcome vitamin A deficiency, it was proposed to genetically manipulate rice to produce beta carotene in the rice endosperm. The presence of beta carotene in the rice gives a characteristic yellow or orange color. Hence, the provitamin A rich rice are appropriately considered as golden rice. The biosynthetic pathway for the formation of beta carotene is now given. The genetic manipulation to produce golden rice required the introduction of three genes encoding the enzymes phytoin synthase, carotene desaturase, and lycopene beta cyclase. It took about 7 years to insert three genes for developing golden rice. Golden rice has met almost all the objections raised by the opponent of genetically modified foods. However, many people are still against the large-scale production of golden rice as this will open door to entry of many other genetically modified foods. Another arguments put forth against the consumption of golden rice is that it can supply only about 20% of daily requirement of vitamin A, but the proponents justify that since rice is a part of the mixed diet consumed the along with many other foods, the contribution of provitamin A through golden rice is quite substantial. Recently, in 2004, a group of British scientists have been developed an improved version of golden rice. The new strain golden rice 2 contains more than 20 times the amount of provitamin A than its predecessors. It is claimed that the daily consumption of 70 gram rice can meet the recommended dietary allowance for vitamin A. Let's know about genetic engineering to increase vitamins and minerals. Transgenic rice developed with high provitamin A content we discussed. Transgenic crop plants are also being developed for increased production of other vitamins and minerals. A transgenic Arabidopsis thaliana that can produce tenfold higher vitamin E, that is alpha tocopherol, than the native plant has been developed. This was done by a novel approach. A thaliana possesses the biochemical machinery to produce a compound close in structure to alpha tocopherol. A gene that can finally produce alpha tocopherol is also present but is not expressed. This dormant gene was activated by inserting a regular gene from a bacterium. This resulted in an efficient production of vitamin E. Some workers are trying to increase the mineral contents of edible plants by enhancing by ability to absorb from the soil. Some success has been reported with regard to increased concentration of iron. Now let's know about commercial transgenic crop plants. The very purpose of production of transgenic plants is for their commercial importance with high productivity. It was in 1995 to 96 transgenic plants like potato and cotton were for the first time made available to farmers in USA. By the year 1998 to 99, five major transgenic crops like cotton, maize, soybean, and canola and potato were in widespread use. They accounted for about 75% for the total 75% of the total area planted by crops in USA. These plants are carefully designed to give rise to products which will improve human health and increase of crop yield. Let's know about the goals of biotechnological improvements in crops. There are about 30 to 40 crops that have been genetically modified and many more are being added. However, very few of them have got the clearance for commercial use. The ultimate goals of genetically modified crop plants now we'll discuss. First is resistant to diseases like insect microorganisms. Second is improved nitrogen fixing ability. Third is higher yielding capacity. Fourth is resistant to drought and soil salinity. Fifth is better nutritional properties. Sixth is improved storage quality. Then production of pharmaceutical important compounds. Absence of allergens. Modified sensory attributes, example, increased sweetness as in thaumatin. Concerns about transgenic plants, the fears about harmful environmental and hazardous health effects of transgenic plants still exists, despite the fact that there have been no reports so far in this regard. The transfer of almost all the transgenic plants from the laboratory to the crop fields is invariably associated with legal and regulatory hurdles, besides the social and economical concerns. The major concern expressed by public is the development of resistant genes in insects, generation of super weeds, etc. Several remedies measures are advocated to overcome these problems. The farmers in developing countries are much worried about the seed termination technology, which forces them to buy seeds for every new crop. These farmers are traditionally habituated to use the seeds from the previous crop, which is now not possible due to seed termination terminator technology. Now let's know about the transgenic plants as bioreactors. Another important application of genetically transformed plants is their utility as bioreactors to produce a wide range of metabolic and industrial products. These aspects now we'll discuss in different chapter, in next chapter. Okay. So any questions regarding the transgenic plant you can ask me in the comment box. I will definitely answer the question and to know all four uh, you can see all four videos of these transgenic plants in my channel and subscribe my channel for to know more updates about biotechnology, new latest technologies, and thank you.